Hi students, welcome to our rapid discussion session. I am Sulekha. Today I am going to discuss the second chapter of the second year portion, that is sexual reproduction in flowering plants. See, the chapter is very lengthy and gives detailed description of the process of sexual reproduction. Now let us see, what is a flower? You have studied that flower is a modified shoot. The shoot meristem undergoes a number of changes to form the flower meristem, and typical flower has got 4 different parts. Okay. So flower is the reproductive structures in angiosperms. A typical flower has got 4 structures such as, or 4 whorls such as calyx, corolla, androecium and gynoecium. Okay. A typical flower has got 4 whorls, okay, such as from outer to inner, one calyx, the second one corolla, the third one is androecium, and the fourth one is gynoecium. Okay, so from outer to inner, these are the 4 different whorls of a typical flower: calyx, corolla, androecium, and gynoecium. The first and second are known as non-essential whorls. Whereas third and fourth are called essential whorls. Because these two are involved in the process of reproduction. Androecium is the male reproductive structure. This is the male reproductive structure, and gynoecium is the female reproductive structure. Okay, female reproductive structure. Androecium has got 2 parts, and its units are called as stamen. A typical stamen has got 2 parts. One is the sterile stalk, that is called a filament. And at the tip of the filament, we can see a box-like structure, and that is called an anther, within which the pollen grains are produced. Pollen grains are produced. And the next one, that is the pistil or the gynoecium, that is divisible into 3 parts. This is a female reproductive structure. The lower swollen part is called ovary, and the tubular structure is called style. Ends with a receptive part, and that is called a stigma. Stigma. Okay. Now, first of all, see how the pollen grains are produced within the anther of the male reproductive structure, that is stamen. Okay. So, the first, now we are going to details of the process of sexual reproduction in flowering plants. Okay. Right. Most of the flowers are bisexual, having both the male and female reproductive structures in the same flower. So now first to see how the pollen grains are produced within the anthers of the male reproductive structure, that is stamen. And before that, we have to see the structure of a typical anther. What is the structure of a typical anther? A typical anther, this is the stamen with the filament and anther. So typical anther is 2-lobed. It is 2-lobed. Anther. It is 2-lobed. then dithecus, and tetrasporangiate. Tetrasporangiate. This two-lobed, and then dithecus and tetrasporangiate. That means if we, these are the two lobes, and if we take the cross section of the right bilobed or two-lobed anther, we can see 4 microsporangia like this. This is the first microsporangium, and second one, third one, then fourth one. Okay, so together we can see 4 microsporangia within a typical anther. Hence, it is described as a tetrasporangiate structure. Okay. Now, let us see how this type of anther develops. That is the ontogeny of the anther or development of the anther and its structure at maturity. In the beginning, it appears as a homogeneous mass of cells like this, and delimited externally by epidermis. And slowly, it becomes a four-lobed one. Okay. And this is followed by the development of a typical anther. Okay. So this finally it becomes a two-lobed one, each lobe having each lobe having single microsporangium. So together in a mature anther we can see 4 microsporangia. So let us see the number of the different types of and the different types of wall layers seen in a mature anther. What are the different types of wall layers seen in a mature anther? I'm going to enlarge incl lobe. This is the outermost layer, that is called epidermis. Okay, wall layers of anther. Of an anther. So outermost is epidermis. It is single layer and provides protection. Inner to this we can see a radially elongated layer. Cells are radially elongated like this, and this is called endothecium, followed by 2 to 3 layers of narrow and thin-walled cells. Like this. Okay. And these layers are called middle layers. And this is followed by the innermost layer, and that is called tapetum. Okay, this is the innermost layer. Like this. And this encloses porogenous tissue in a young or developing anther. So within this we can see the porogenous tissue. Like this. It's completely filled with cells like this. Let us label the various layers. What do you call this? This is the outermost layer and it is called epidermis. And the next one is called endothecium. Followed by 2 to 3 layers of thin-walled and narrow cells, and it forms middle layers. And then comes the innermost layer of anther wall, and it is called tapetum. And inside this we can see the porogenous tissue. Porogenous tissue, okay. And this is right, various wall layers of anther and single lobe enlarged. Single lobe enlarged. And see here, out of the various wall layers, it is tapetum that completely surrounds the inner sporogenous tissue. Okay. So function wise, this is mainly concerned with the transport of food materials into the sporogenous tissue. So tapetum is a special mention, and it is nutritive in function. Nutritive in function. Okay. This is fertile tissue. So if we take the cross section of an anther, we can clearly see the four lobes with all layers of anther. So in cross section, it appears like this. The sectional view, okay. So transverse section or cross section of an anther. In the center, we can see the connective that connects the two anther lobes together with the various wall layers. Epidermis is followed by endothelium, like this. And then we can see the middle layers starting from here, ending here. Two to three layers are seen like this. Okay. Followed by the innermost layer, that is tapetum, like this, having sporogenous tissue. See why this endothelium and middle layers run from this point and ends here in all the four lobes. And this is the position of thin-walled cells. Here we can see thin-walled cells called a stomium. And that marks the point where the anther dehisces to release the pollen grains. So this is the position of the thin-walled cells called a stomium, where the anther breaks to release the pollen grains on maturity. Okay, so this is the sectional view of a developing anther, and this is the sterile tissue that is called a connective, which connects the two anther lobes together. And look into this tapetum here. It is multinucleate. You can see more than one nucleus in the tapetal cells. So the multinucleate condition is because of endomitosis. Of course, there is a formation of daughter nuclei, but it is not, karyokinesis is not followed by cytokinesis. Hence, the number of nuclei per cell increases. Okay, so multinucleate condition is common in the tapetal cells. Now let us see what happens to the porogenous tissue on maturation. When the anther matures, we can see some changes occurring in the porogenous tissue. So in the beginning, all the cells are interconnected by plasmodesmatal connections. And later the plasmodesmatal connections get replaced by wide cytoplasmic channel. And finally, the cytoplasmic channels also get cut off, and each cell comes to life freely within that anther locule, and now they are known as microspore mother cells. Okay, so these are the various changes that we see in the sporogenous tissue as it grows. So in young anther, right in the center, we can see here, we can see the sporogenous tissue. Okay, right, and later the cells get separated from one another, and each cell will develop into spore mother cells. Mother cells. That is microspore mother cells. Microspore mother cells. Okay. And what is the ploidy of the microspore mother cell? The sporogenous tissue is 2N because it's a part of anther. It's a part of the flower. Flower is a part of sporophyte. And in higher plants, sporophyte is 2N. Right? So the sporogenous tissue is 2N and the spore mother cells are also 2N. In the previous chapter, you have studied that during gametogenesis in higher plants, right, meiosis undergoes. Okay, the way by which the gametes are produced is through meiosis. So the mother cells will undergo the reduction division, that is called meiosis. So the spore mother cells, in short, they are written as spore mother cells, undergo meiosis. That is reduction division, and meiosis result in the formation of 4 haploid spores. This is the spore mother cell. So the resulting spores are called microspores. So it results in the formation of 4 microspores. The resulting microspores get arranged variously. We can see different types of microspore tetrads. It may be linear, T-shaped, inverted T-shaped, tetrahedral, isobilateral, and decussate. So different types of microspore tetrads we can see in anther. Of that, the most common one is called isobilateral. This is an isobilateral tetrad with 4 microspores, and all the spores are held together by a common outer structure, and that is called a callus. The binding structure is called a callus, and each cell is known as microspore. Ploidy is N. So thus, from diploid microspore mother cell through meiosis, there is formation of 4 haploid microspores. And soon after, this meiosis, that is a reduction division, the resulting microspores are held together by a common outer callus. Callus is the binding material. Okay? And these cells will remain within the callus till each microspore completes its further development. That means during this period, we can see the microspore synthesizing the wall layers such as intine and exine. And once intine and exine synthesis is over, the callus is degraded enzymatically and the microspores are released into the anther locular anther cavity. And such mature microspores are known as pollen grains. Okay. So they undergo development, then the callus get degraded and all the spores are made free now. They come to life freely within the anther locule. Okay. And these are known as pollen grains. Pollen grains. What are pollen grains? Mature microspores are known as pollen grains. Okay. And this we can see hundreds of pollen grains within the anther locule or anther cavity of the anther. Okay. Hundreds of pollen grains we can see. Now, what is the structure of a typical pollen grain? Structure of a typical pollen grain. structure of a typical pollen grain. A typical pollen grain has got, this is the surface view. We can see ornamentation or sculpturing pattern, and here and there thin areas are seen, and these are called germ pores. So this is surface view. This is the germ pore, and this is the outer layer, that is exile. Whereas in sectional view, right, it shows its contents in a detailed manner. Sectional view, right, it shows it is exile with the sculpturing pattern, and inner to this we can say the intine with its nucleus and contents. Okay, so the typical, right, pollen grain has got two layers, and they are, this is exile and the inner one is called intine, and this is pollen nucleus. Having its plasma membrane. Okay, exine is very thick, and it is composed of a special material that is called sporopollenin. Sporopollenin, and it is resistant to, right, physical factors as well as, right, chemicals. Hence, right, pollen grains can be easily preserved in fossil. So it is having a characteristic material that is called sporopollenin. So this is the detailed structure of a pollen. Now, on maturity, the two microsporangia, or the adjacent two microsporangia of each anther lobe fuse together to form a horseshoe-shaped structure, and at the point of stomium marks where it breaks to release the pollen grains. Okay, we have already studied that. And at the junction of the two microsporangia of each anther lobe, thin-walled cells called the stomium we can see, and the stomium, right, marks the point where, right, the anther breaks to release the pollen grains. So when the, right, the pollen grains mature, right, the anther dehisces and releases the pollen grain. And before anther dehiscence and release, we can see some more changes taking place in this pollen grain. And what's the ploidy of pollen grain? Of course, it is haploid, because this is a mature microspore. Now let us see, right, what changes occur in the pollen grain before the anther dehisces, right, for the release of pollen grains. This is the uninucleate, and this haploid nucleus is in the center. Now, in the next stage of development, we can see this centrally located nucleus getting shifted towards one side, and that marks the future position where the generative cell is going to develop within this pollen grain. So, right, the nucleus gets shifted towards one side. So, next is development of pollen grain. Pollen grain. This is explained with the sculpturing patterns. This is intine. Now the nucleus gets shifted here, right? Then it divides mitotically. The second nucleus gets shifted here, and this is followed by the plasma membrane like this. Now the plasma membrane will break here. and that grows over, right, one of the nuclei here, okay? And later the whole thing gets pinched off from the plasma membrane, and that gets released into the cytoplasm of the parent pollen grain, and thus there is formation of a small cell within the parent pollen grain, and the newly developed one is known as the generative cell. Now within this we can see the formation of a small cell, and that is called a generative cell. So after the formation of the generative cell, the pollen grain will appear like this. Yes. Now we can see the intine, and within this there is formation of a new cell, and this spherical cell is the generative cell. This newly formed cell is known as the generative cell. So the parent pollen grain now, right, consists of a new, right, spherical cell, newly formed spherical cell, and this is called the generative cell. Okay, in this stage the pollen grain is referred as two-celled. So this is known as two-celled pollen grain. In more than 60% of angiosperm, the anther dehisces to release the pollen grains when it is two-celled. Okay. Now we can see the release of pollen grains from the mature anther. And later, right, it becomes three-celled. In certain other plants, at the time of dehiscence of the anther, it will be three-celled. Three-celled means the nucleus of the generative cell divides mitotically, and there is formation of the two sperms or the male gametes, and thus the pollen grain becomes three-celled. So this is two-celled, and three-celled pollen grain will appear like this. Okay. This is a three-celled pollen grain. Pollen grain, and these two represent the, right, male gametes. Okay. So the anther breaks to release the pollen grain either in the two-celled stage or three-celled stage, and in majority of angiosperm, right, the anther dehisces to release the pollen grains at two-celled stage. Okay, so this is all about the microsporogenesis. So what do you call the process by which microspores are produced within the anther? That process is called microsporogenesis. Microsporogenesis refers to the formation of pollen grains within the anther. And mature microspores are known as pollen grains. Mature microspores are called pollen grains. Okay, and the dehisces to release the pollen grains at two-celled or three-celled stage, and the dehiscence of the anther occurs, right, mainly at the junction of the two microsporangia of each anther lobe, where we can see thin walled cells called, right, stomium. Rarely we can see the tip of this anther, right, breaking and releasing the pollen grains through pores. Now, let us see, right, the importance of this pollen grains. Pollen grains, right, honey, you know, what is honey? That is the flowers are having nectar, and it is taken by the honeybees, right, and later it is converted into, right, honey, and within the honey we can see pollen grains, right, and it is actually a supplement for food rich in nutrients like vitamins, then you can see different types of sugars, minerals, etc. And now it is available in the form of either capsule or pollen syrup, right. So it has got high nutritive value. And at the same time, we can see certain pollens of plants causing some sort of allergy, right, to us. And this is because of the proteins present in the pollen grains. Pollen grains are rich in protein also. In some people, the pollen grains can bring about some, right, allergy, right. That is asthma problems, bronchial problems, respiratory problems like that. This mainly because of the different types of proteins present in the pollen grain. A typical example for, right, a pollen causing some sort of allergy is the pollen grains of Parthenium, that is called a carrot grass. Also called, that is Parthenium. Scientific name, right, genus name Parthenium, commonly known as carrot grass. So with this, microsporogenesis is over. Now let us see how the megaspores as well as the female gametophyte or embryo sac is developed within the ovule. And for that we have to go back to the female reproductive structure called a pistil, right, or gynoecium. What is the structure of the pistil? The next one. Structure and development of ovule and female gametophyte. The female reproductive structure is known as the gynoecium. It has got three parts: the swollen basal part, then the tubular style, and the receptive stigma. Okay. And within the ovary, we can see carpels, chambers like this, and inside that, spherical structures called ovules are seen. So within the ovary, spherical structures called ovules are seen. So ovary with ovule, this is the style, and this is the receptive part, that is called stigma. Okay. Now let us see the structure of ovule. A typical ovule has got a massive tissue in the center. Massive tissue in the center, and that is called a nucellus. This nucellus is covered and protected by means of usually two protective coverings called inner integument and outer integument. Okay, so these are the two, right, protective coverings called integuments. This is outer integument and this is inner integument. Okay, the massive tissue is called nucellus and the small stalk is called funiculus. The point of attachment of the stalk, that is funiculus, with the body of the ovule marks hilum. And look into these integuments. These integuments will not cover the entire nucellus, but leaves a small opening towards one side, and that opening is called micropyle. This opening is called micropyle. That region is called a micropylar region, and the opposite side is called chalazal side or chalasa. So micropylar side and chalazal side, or micropyle and chalasa are the two different parts. Clear? And the micropyle is actually left for the embryo pollen tube right into the embryo sac to bring about fertilization. So this is the structure of a typical ovule, and this type of ovule is known as anatropous or inverted. And see, this is the most common type of ovule in angiosperm. Several types of ovules we can see, of which the most common one is inverted one, that is anatropous. And another one is called erect one, like this. This is another type of ovule, and it is called orthotropous. Okay, an erect one, whereas here it is inverted. So based on the position of micropyle with respect to funiculus, in angiosperms we can see different types of ovules. Of course, I have drawn the diagrams of two types of ovules here. They are anatropous and orthotropous. So anatropous, orthotropous, then we can see campylotropous. So this is the most common, the second type. The third is called campylotropous. That is the third type. The fourth one is called hemianatropous. And the fifth one is called amphitropous. Amphitropous. Right? So different types of ovules we can see, and this is based on the position of micropyle with respect to funiculus. Now for the family Cactaceae, right, there's a special type of ovule, and that is called a cercinotropous. Remember, this is not a sixth type, but it is unique to the family Cactaceae. So for family Cactaceae, there is a unique type of ovule, and it is called cercinotropous. Seen in members of the family Cactaceae. So this is the structure of a typical ovule. So we can see two protective coverings called integuments. They are inner integument and the outer integuments, right? And such ovules are called bitegmic. Ovules with two integuments are called bitegmic. Ovules with a single integument, it's a rare condition, unitegmics. And again, rarely we can see ovules without any integument, and they are called ategmic. But the most common one is bitegmic with outer and inner integuments. So this is the structure of the ovule. The next one is megasporogenesis. That is formation of megaspore mother cell, and followed by megagametogenesis. That is the formation of the female gametophyte or embryo sac. So first we can see megasporogenesis. Is the process by which megaspores are formed from megaspore mother cell. So it refers to formation of megaspores from megaspore mother cell. a megaspore mother cell. Okay, and for that, once again we have to go back to the ovule. This is nucellus with the two integuments like this. Okay. Nucellus is a massive tissue with hundreds of cells. The whole thing is diploid. Now, one of the cells of nucellus situated towards the micropylar side of the ovule. We can label this as that cell. Okay? And this cell is known as megaspore mother cell. Now, one of the cells of nucellus situated towards the micropylar region, right, of the ovule becomes, right, gets enlarged, right, and it appears, right, an enlarged cell, possesses dense muscle cytoplasm and a prominent nucleus. And this conspicuous cell is known as megaspore mother cell. Can you tell me the ploidy of the megaspore mother cell? See, the entire ovule is 2n. So the cell also will be 2n. So the megaspore mother cell is 2n. Hence, during megasporogenesis, like microsporogenesis you have studied taking place in the anther, and here also the megaspore mother cell undergoes meiosis, that is reduction division. So the megaspore mother cell undergoes meiosis, and there is formation of 4 megaspores. The resulting megaspores get arranged in a linear fashion alone, whereas the microspores get arranged variously, that you have studied now. So here the megaspores get arranged in a linear fashion. That is 1, 2, 3, 4, like this. Thus it appears like this. The nucellus with the integuments, and here we can see the megaspores. This is the linear row of megaspores. So out of this, normally only one of the megaspores undergo the rest of the division, okay, rest of the processes, whereas the remaining 3 will degenerate and disappear. Okay, so usually the cell one remains functional and rest of the 3 micropylar will degenerate. So let us take it out. The whole thing I am going to take out, and this is the functional one, and these are the degenerating megaspores. So only one remains functional, whereas these 3 will degenerate. So this is the functional megaspore, and we can label the rest of the 3 as degenerating megaspores. Okay. Now what happens to the functional megaspore? What do you call this process? This process is called a megasporogenesis. This is megasporogenesis. And next one is megagametogenesis, means development of the female gametophyte. This is followed by megagametogenesis. gametogenesis. Okay. Process begins with the functional megaspore. So what's the nature of this? Or it is haploid, because the a division by which the megaspores are formed is meiosis, that is reduction division. So it begins with the enlargement of the functional megaspore. It is uninucleate in the beginning. Now we can see this undergoing 3 repeated mitotic divisions. 3 repeated mitotic division. Okay. Nucleus undergoes first division, that is mitosis 1. It is over. Followed by second and third division, mitotic division. Undergoes the second division, and the resulting nuclei we can see getting pushed to the poles, micropylar and the chelacal pole. Right, that is, so this is the functional megaspore. This mitosis 1, mitosis 2, followed by the third mitosis. It is mitosis 3. By third division, we can see 2 groups of 4 nuclei getting organized in the micropylar end and chelacal end. Okay. Nuclear divisions are over, and so far the we cannot see cellularization till the third division is over, and this is followed by rearrangement of the nuclei and wall formation to form the female gametophyte or embryo sac. From each group, one gets pushed to the center, right? And they act as the polar nuclei. This is followed by wall formation around all the nuclei of this micropylar and chelacal side, and that result in the formation of 3 cells in the chelacal side like this, and another group of 3 cells towards the micropylar end. Okay. And the 3 cells of, this is the chelacal side, and this is micropylar side. These 3 cells are known as antipodals. The large cell is the central cell, and these are the 2 polar nuclei. The 3 cells seen towards the micropylar side is together called agapparatus. The 2 laterally placed cells are called synergids, and this cell is called egg cell. Okay. This 7-celled and 8 nucleate structure is known as the female gametophyte or embryo sac. So this, in angiosperm, the embryo sac or the female gametophyte is described as a 7-celled and 8 nucleate structure. Okay. This is the most common type of embryo sac development in angiosperm. And in this type of embryo sac development, The functional megaspore is telasel one. It is monosporic, uninucleate, and the mature embryo sac is 7-celled and 8-nucleate. And this type of embryo sac is known as polygonum type of embryo sac. What do you call it as? Polygonum type. This is called polygonum type of embryo sac. Okay. So this we have completed both the microsporogenesis and megasporogenesis. You have already studied the structure of the mature pollen grain and a female gametophyte or embryo sac. So on one side we can see the release of pollen grains either in the 2-celled or 3-celled stage, and in the other side the ovary is having ovule. Within the ovule we can see the formation of the so-called 7-celled and 8-nucleate structure, which is known as embryo sac or female gametophyte. Okay. So next one is pollination. What is pollination? Pollination is the process by which pollen grains are transferred from the mature anther to the stigma of the pistil to bring about fertilization and seed formation. The next is pollination. The process by which pollen grains are transferred to the stigma. The process by which pollen grains are transferred to the stigma. That process is called pollination. Right. Pollination may be of two types. One, self-pollination, and the second one is called cross-pollination. In self-pollination, pollen grains are transferred from the anther to the stigma of the same flower. Whereas in cross-pollination, we can see transfer pollen grains from one flower to the stigma of another flower. It can be between two varieties, two species, and rarely two genera. Nature prefers cross-pollination over self-pollination because cross-pollination can bring about variation in the resulting progeny. Remember, such variations are very important for a process to occur in nature, and that is called, what do you call that process? It is evolution. So nature prefers cross-pollination over self-pollination. And again, based on the nature of the pollinating agent, pollination, of course, require the involvement of pollinating agents, especially for this cross-pollination. Okay, so different types of pollinating agents we can see, and based on that, the pollinating agents may be, can be further divided into two types. That is based on the pollinator or pollinating agents. Pollination may be of two types, abiotic and biotic. Abiotic, one wind, the other one water. The agents are wind and water. And then biotic, based on the nature of the pollinating agent, different types we can see. We can see, in most of the cases, the pollinating agents are insect. That is called entomophily. Insect pollination is called entomophily. Where the pollinating agents are insects. Then we can say, right, birds, then sometimes bat, snails, ants, like that. Accordingly, right, the name varies. Okay. So different types are there. I think we need not go too much details to that. Okay, different types of pollinating agents. The most important one is called right entomophily, where the pollinating agents are insects, and of that the main one is honeybee. Yes, honeybees. Okay. Now let us see some adaptations, right, shown by plants having particular type of right a pollination method, that is wind, water, and insects. And what are the common adaptations that we see in such plants? So common adaptations. Adaptations. Right. Wind pollinated flowers, small in size, mostly grouped in or produced in inflorescence groups. Okay. Then the corolla, notch colored, without, flower is without honey. But we can see certain other adaptations for right the gynoecium and the androecium. Ovary, it is single chambered, small style with trifid stigma. This is the ovary with a single ovule and feathery stigma. Okay. Feathery stigma. Okay. And similarly, the stamens will show long filament like this, and the anthers are placed perpendicular to this. And this type of stamens are called versatile. They can move freely in air currents. Okay. And also, right, this adaptation of the pistil enable the pollen grains to trap inside this, the hairy stigma like this. So these are certain common adaptations seen in wind pollinated, right, plants. The second one, water pollinated. Water. See, all hydrophytes are water plants are not pollinated by water. Okay. And this is water pollination is seen in right hydrophytes that are submerged in water. Right, seen totally inside within this water, submerged ones. But we can see Iconia you have studied. It's a floating one. Pistia, it's a floating one. So this is commonly seen in right plants that are submerged in water. Example, Vallisneria. Vallisneria. Then marine grass is there, that is called Nostera. It's a marine grass. And the most important adaptations in water pollinated right plants is the pollen grains are so long and ribbon shaped pollen grains, long and ribbon shaped like this, having a mucilaginous outer covering. The mucilaginous covering, pollen grain with mucilaginous outer covering which protect the pollen grain from getting wet. Outer covering. Vallisneria is dioecious. Right, we can see separate, right, plants, male and female plants. And when the flowers mature in the male plants, they get detached and the flowers are taken to the surface of the water. We can see the male flowers floating on water surface, and also right in the immediate water we can see pollen grains. When the female flowers mature, the female flowers are having long stalk, pedicel, okay? And usually it is seen in a coiled condition. And as a result of coiling, it is taken, the flowers, female flowers are taken towards the water surface, right, and it will remain as such for few days. By that time pollination will be over, and slowly, right, the stalk will start coiling and the flower is, female flower is taken inside the water, and the rest of the processes occur within the water, right. It is very nice to see how the process of pollination occurs in such a submerged plants like Vallisneria. The next one is, right, the adaptation seen in, right, insect pollinated one. Insect pollinated plants. The first one is showy flower with colored corolla. Showy flower with colored corolla. Okay. And the second character, presence of nectar. Right. And the third, pollen grains will be colored and sticky. Colored and sticky pollen grains. The sticky nature is due to lipid, and color is due to carotenoids, okay, that enable the pollen grains to get attached in the body of the pollinator. And in night, flowers that opens during night, the corolla will be white in color mostly, and also there will be, right, a smell, okay, which will enable the pollinator to move towards the plant. And this is certain adaptation commonly seen in, right, insect pollinated plants. Now, based on the source of pollen grain, we can see, right, different types of pollination. And before that, let us go to the two types of flowers. Most of the flowers, right, open on maturity and exposes the floral whorls for pollination. Whereas in certain others, right, even after maturity, it will remain in a bud condition. So based on that, right, there are two types of flowers. One is called, right, chasmogamous, and second one is called clistogamous. Types of flowers. Chasmogamous. This is the most common type. Common type. And second is called clistogamous. What are clistogamous flowers? Right, even after maturity, it remains in a bud condition. So remains in bud condition. Even after maturity, right? Examples are Commalina, oxalis, and viola. Okay, these are examples for listogamous flowers. Now next, based on source of pollen grain, we can see three types of fertilization. So based on source of pollen grain. We can see three types of pollination. And there, one is called gittanogamy. Okay, the first one is called gittanogamy. Okay, what is gittanogamy? Gittanogamy, first one, okay, or we can mention the first one, that is autogamy. Autogamy, next gittanogamy, and the third one is called xenogamy or allogamy. The autogamy, that is self-pollination. Pollen grains are transferred from the anther to the stigma of the same flower, right? And then in gittanogamy, pollen grains are transferred from the anther to the stigma of another flower of the same plant. Another flower of the same plant, right? That is gittanogamy. So actually it requires, right, a pollinating agent, but genetically there is no difference. Then last one, xenogamy, right, and here, right, it is cross pollination. This is cross pollination. And nature actually prefers cross pollination over self pollination, just because cross pollination can bring about variation, that is changes in the resulting progeny. Accordingly, we can see several adaptations in plants, that is for preventing selfing and promoting crossing, right? Known as outbreeding devices. Outbreeding devices. The first one, unisexuality. The second one, self-sterility. And the third is heterostyle. The fourth one, dichogamy, means maturation of male and female reproductive structures at different times, right? It may be of protandry or protogyny. Protandry means the male structure matures first. The male reproductive structure matures first. For us here, female reproductive structure matures first. Okay. Unisexuality, you know, formation of unisexual flowers. Self-sterility, right, due to some reasons, right, self-fertilization is prevented. Then heterostyle, male and female reproductive structures are placed at different levels. Then next one, right, that is dichogamy. That is maturation of the male and female reproductive structures at different time, different period. And one more is there, that is ergogamy. Through some, right, mechanical means, due to some structural adaptations, self-pollination is prevented. So due to structural adaptations, selfing is prevented. Okay, selfing is prevented. This ergogamy. Example, calotropis. With gynostegium. And also we can see another adaptation, right, in the anthers of family Asclepidaceae, where this calotropis is coming. In Malayalam it is called irikku. And here, right, all the pollen grains of each anther lobe fuse together to form a pollen mass, that is called pollinia, a bag-like structure we can see. This is the structure with a small stalk-like structure at the tip. Right. The sac is seen with the pollen grains. It is pollinia. All pollen grains of each anther lobe will fuse together to form a, right, a common sac-like structure, pollinium, that is singular, and together it is called a pollinia. And a sticky structure we can see, right, that is corpusculum. The small, right, tail-like structure is called a caudicle. Together it is called a translator. Translator. So translator mechanism of pollination is common in members of the family Asclepidaceae. Typical example is calotropis. Okay. So this is all about, right, pollination, right, the pollinating agents, the different adaptations we see in, right, different types of plants to mate with a particular type of pollination, right, etc. The next one, right, how can we do this cross pollination, right, by the breeder, that is artificial pollination and its steps. And you have already studied that nature prefers cross pollination over self pollination, because cross pollination can bring about a variation, right, in the resulting progeny, right. So in nature, right, we can see cross pollination due to certain adaptations, right, that prevents selfing and promotes cross pollination. We can also do that process artificially, and that is called artificial, right, hybridization, and it is done by a breeder, right, with a view to develop, right, a desirable hybrid. Means hybrid having both the desirable qualities of the parents used in the hybridization process. Okay. So if we do that, right, by a breeder, it is called artificial hybridization, but in nature it occurs through a process called a cross, right, pollination. So artificial hybridization and different steps. The first step, different steps. Okay. The very first one is the selection of parents. So one, selection of parents. It should be done on the basis of certain desirable qualities. For example, high yield, disease resistance, like that. So based on this, selected two parents. Okay. And next one. So we have selected one as the female parent and the other one as male parent. And both may be having bisexual flowers. Here also we can see bisexual flowers. So next step is removal of the anthers from the flowers of the parent that is selected as the female one. Otherwise, there is chance for cross-pollination. Okay. So next one is the emasculation. Emasculation is removal of anther. Removal of anthers from the bisexual flowers of female parent. Otherwise, chance, right, there is chance for self-pollination. So second step is over. The third one, after emasculation, we have to put a bag over this, right, female flower from which the anthers are removed. That process is called bagging. Bagging. By this time, right, the male flowers will mature, right, that is the flowers of the male parent will mature, right, and it is time for to collect the pollen grains from the anthers of the male parent. So just collect the pollen grains with the help of a fine camel hair brush, right, it should be done by the breeder itself, and then just right, put it over the stigma of the right, female parent. So pollination is done by the breeder itself. The next one, pollination done by breeder itself. And soon after pollination, we have to put the again over the pollinated flower. So next one, rebagging. What's the need of rebagging? Pollination is already over. Now, what is the need of putting the bag once again? See, the pistil can receive pollen grains from any other outside plant as long as, right, it remains in a receptive condition. And when it loses the receptivity, the stigma becomes dark in color. So by that time, we can remove the bag. Until that, right, we should keep the bag over the pollinated flower. So rebagging, right, and next one, by the time, right, after few days, we can see the stigma becoming dark in color, and it is an indication that stigma has lost its viability, and by that time, we can remove this, right, bag permanently, allow the plant to, right, undergo fertilization, then the fruit formation and seed setting, followed by seed setting, right, and this, collect the seeds and raise the next generation. So collect the seeds, and raise the next generation. And such resulting plants are called hybrids. Hybrids show better performance than the parents, and the vigor and the vitality shown by these hybrids over their parents is called heterosis or hybrid vigor. So these are the various steps, right, the breeder has to follow to do artificial hybridization. Now let us go back and see how the process of fertilization occurs. Okay. So we have studied, right, the microsporogenesis, megasporogenesis, pollination, different types, pollinated factors, right, and importance of cross-pollination over self-pollination, and also how this artificial hybridization can be done by breeder in the breeding institution. Now let us see what is the fate of the pollen grains once it gets collected in the stigma of the pistil. So next is pollen pistil interaction. Pollen pistil interaction. After pollination, we can see some reaction occurring between the proteins of the pollen and the stigma to see whether that particular pollen is matching, means compatible or not, matching or not, compatible or non-compatible. So that is through a reaction between proteins of the pollen and that of the stigma, and it is the beginning of the pollen pistil interaction. Suppose the pollen grains are found to be compatible, matching, the pollen grains can absorb sugary substances, as well as moisture from the stigmatic surface. The size of the pollen grain increases many times. Exine ruptures and the intine along with the contents will flow out through the thin areas called germ pores and result in the formation of a tubular structure called pollen tube. The process is called pollen germination. So pollen germination, this is the ovary, style and the stigma. Here we can see the pollen grains getting collected. Pollen grains. After pollen pistil interaction, if it is found to be compatible, it can germinate. So pollen grains germinate if they are found to be compatible. This is exine. This is intine. It will come out in the form of a small tube that is called a pollen tube. The process is called pollen germination. Pollen germination, it carries the male gametes. Also called as sperms, and here we can see the contents. The whole thing will flow into the pollen tube. So this is pollen germination, and this tubular structure is known as the pollen tube. Okay, so the pollen grain now grows down by piercing the cells of the style, to reach the ovule for fertilization. And then based on the nature of the style, we can see two types of style. We can see one is called solid and the other one is called hollow. In solid type of style, the pollen grains has to pierce each and every cell of the style to go to the lower side where we can see the ovules. The swollen part of the pistil is the ovary. Within that, ovules are seen. So it has to pierce each and every cell of the solid style to reach the ovary and for entering the ovule. Okay. Yes. So two types. One is called a solid type and other one is called the that is the hollow type. That parenchymatous cells that fills the style in solid type is called a transmitting tissue. Transmitting tissue is the parenchyma that fills the entire length of the style. And why the pollen tube carrying the male gametes always grows downward? Why can't it go upward? So far there is no report of pollen grain moving away or out of the style. It always grows downward. And this is because of the release of certain chemicals from a finger-shaped structure seen in the microphylla side of the synergids called filiform apparatus. Okay, it is filiform apparatus that guides the downward growth of the pollen tube towards the ovule for bringing about fertilization. So it grows like that. Now let us see how this fertilization takes place. Let us Go back to the chapter. Yes, it's a sectional view of a typical, right, a bisexual flower. This is the female reproductive structure, the stamens, right, then the petals, sepals, like that. Be familiar with all diagrams given in the text. This is a typical, right, stamen with a long filament and anther. The sectional view, right, the tip of the anther is removed. And here we can see the, right, this is the tetrasporangiate nature. These are the two adjacent microsporangia of each anther lobe, right, pollen grains. And this line marks the line of dehiscence. And here we can see the thin-walled cells called the stomium. This you have studied in detail, right, the various wall layers of an anther and how it appears on maturity. So here it breaks. That is the marks the point of thin-walled cells called the stomium. And also by that time we can see the fusion between the two adjacent microsporangia of each anther lobe. They fuse together to form something like a horseshoe-shaped structure. And it breaks here where we can see the thin-walled cells called the stomium. This is the, right, ultrastructure of pollen grains. The photos taken through scanning electron microscope. It clearly shows the exine pattern. And this explains the formation of generative cell within the parent pollen grain. Everything you have studied in detail. Yes. Then the pollen products, they are rich in nutrients. Okay. This is the first one. This is the, right, gynoecium of hibiscus. Hibiscus, okay. Ovary, style and stigma. It is pentamerous, having 5 carpels and 5 stigmatic lobes. And this is power poppy. Right. It is multicarpellary and syncarpous. Multicarpellary and syncarpous. And this is, right, mycelia. That is Malayalam. It is called a chembagam. And each structure represents carpel. It is multicarpellary and apocarpous. Apocarpous means free carpels. And this is the, right, view of the sectional view of a typical ovule, and it is anatropous. The process of megasporogenesis, megaspore mother cell, right, it is 2N, undergoes meiosis. There is formation of 4 megaspores. Only one remains functional, that is the chelacal one, undergoes 3 repeated mitosis, and finally there is formation of, right, a 7-celled and 8 nuclear structure called embryo sac. Yes, right, this is, right, a Comelina showing both cleistogamous and chasmogamous flowers. And this plant has got both type of flowers, chasmogamous and cleistogamous. Cleistogamous flowers are seen towards the, right, lower part, that is, which is near the soil. And here it is wind pollination. Okay. And this is cone inflorescence. Trilla zostera, examples for water pollination. And this is Vallisneria, the male flowers, right, and then the female flower. Okay. Now let us see how fertilization occurs. See, this is the sectional view of a pistil, right, showing the downwardly growing pollen tube, ovary, style and stigma. Pollination results in the collection of pollen grains in the stigmatic surface. After pollen pistil interaction, if the pollen grains are found to be compatible, means matching, they can absorb nutrients from the, right, stigmatic surface. The exine breaks and the contents flow out in the form of a small tube, that is pollen tube. Now we can see the pollen tube growing downward, means towards the ovary to enter into the ovule. And based on the mode of entry of pollen tube into the ovule, fertilization may be of 3 types. One is called, right, one is porogamous, second is chalazogamous, and third one, mesogamous, or porogamy, chalazoogamy, and mesogamy. Is it clear? This depends upon, right, the mode of entry of pollen tube into the ovule. So pollen tube first enters into the ovule. Of course, in ovule there is an opening that is called micropyle. Micropyle is meant for the entry of, right, pollen tube into the ovules. Rarely in certain plants we can see a plug or a block in the micropylar side. In such cases, right, the pollen tube will take another pathway to enter into the ovule, and that may be either piercing the cells of the charasa, right, or by piercing the cells of integments. So accordingly, right, the fertilization may be porogamous, where the pollen tube enters through the micropyle. Second one, right, a chalazogamous, where the pollen tube enters into the ovule through the chalasal region. And third one, right, mesogamous, where it penetrates the cells of the integments. And of the three different types, the most common one is porogamy. So here we can see porogamy. It is entering, the pollen tube is entering into the ovule through micropylar region of the ovule. See, whatever be the mode of entry of pollen tube into the ovule, it always enters into the embryo sac, right, through the micropylar region, just because the egg apparatus is placed in the, right, micropylar region. And see this egg apparatus, here we can see the two laterally placed synergids and the central egg cell. So in sectional view, the egg cell is seen behind the laterally placed synergids. And also we can see finger-shaped structures, the micropylar side of the synergid, that is called a filiform apparatus. And this filiform apparatus, right, secretes some chemicals which guides the pollen tube towards the, right, ovule, and finally to enter into the embryo sac. So this is the sectional view of the micropylar side of the embryo sac, and this is the synergid, and this is the egg cell. And here, right, we can see the two polar nuclei. So after, right, pollination, and the two polar nuclei will fuse together to form secondary nucleus, and also out of the two synergids, one will show signs of degeneration, and that is called a degenerating synergid, right, and the other one will remain as such. You see, it is to this degenerating synergid the pollen tube enters to release the contents. So this is the degenerating synergid. The other one we can see on the other side, and this is the egg cell. Now we can see the pollen tube entering into the cytoplasm of the degenerating synergid through the so-called filiform apparatus. It enters into the cytoplasm. The tip of the pollen tube breaks and releases the pollen tube contents. And that discharge mainly consists of the two sperms, then remnants of the vegetative nucleus, and little amount of cytoplasm. Now let us see the fate of the, right, male gametes that get released into the, right, cytoplasm of the degenerating synergid. So one of them will go to the cell, fuses with the egg nucleus, and that is called a syngamy. Syngamy result in the formation of zygote. Zygote will later develop into an embryo. Now what is the fate of the second sperm? That will go to the central cell and fuses with the fusion product of the two polar nuclei called a secondary nucleus, and this fusion is called a triple fusion, just because it involves fusion between three nuclei. Okay, and triple fusion result in the formation of a triploid nucleus called a primary endosperm nucleus. Now the zygote will develop into embryo, and from the, right, primary endosperm nucleus there is formation of the nutritive tissue called endosperm. So thus in angiosperm, endosperm is generally triploid, and it is a post-fertilization product or post-fertilization structure. So this diagram shows, right, the process of syngamy. Okay, let us continue. So to fertilization you have studied. So syngamy. Syngamy is the fusion between, right, one of the sperms and the egg nucleus. Okay, fusion between the sperm and egg nucleus. Okay, result in the formation of zygote that will develop into a multicellular well-organized structure called embryo. And second is triple fusion. Is the fusion between the second sperm and secondary nucleus. Secondary nucleus. This is called triple fusion. This is called triple fusion and results in the formation of a triploid cell. This is 3N and it is called a primary endosperm nucleus. This is the cell. Okay, this is the nucleus. Primary endosperm nucleus, and this is the central cell actually, where the triple fusion occurs. Okay, so that results in the formation of the primary endosperm nucleus. This is 3N triploid, and this will develop into the nutritive tissue that is called into endosperm. Again 3N, the nutritive tissue, right? And this is so characteristic to angiosperm, right? And here we can see two types of fusion, and together it is called a double fertilization, which is characteristic of angiosperm, right? So with this, fertilization is over, and next one, post-fertilization development, that is embryogenesis. You have already studied in the previous chapter now. Okay. Next one, embryogenesis. Refers to the process of development of embryo from zygote. So zygote will develops into a multicellular well-organized structure that is called embryo, and it is called embryo. What's the fundamental difference between the embryo of a dicot and monocot? Right, angiosperm consists of two groups. One is dicotyledons and second one monocotyledons. So what's the difference between embryo of dicots and monocots? Right, this is having two cotyledons. Okay, these two structures are the cotyledons, and this is plumule, and this is radicle. Monocots have single cotyledon, and that cotyledon is usually a massive structure like this, a single cotyledon. Now let us see the process of embryo development in dicots. Okay. So this is zygote. Undergoes predetermined division. It is unequal first. First of all, two cells are formed. This is apical cell designated as CA. This is basal cell, right, that is designated as CB. And the embryo proper is developed from CA, whereas the cell of the CB, the basal cell, will undergo repeated transverse division and there is formation of a tail-like structure, right, that is called a suspensor, by which the embryo proper is attached to the inner side of this, right, seed cavity. So there is formation of CA will undergo repeated division, then there is formation of a globular proembryo like this. Globular proembryo. It becomes 2-celled, 4-celled, 8-celled, right, then 16, 32 like that. And it is continued till, right, there is formation of a globular structure like this. And by that time, this CB will undergo repeated divisions and there is formation of a structure like this. This is the vesicle. Whole thing is called a suspensor. Now we can see there's a globular proembryo getting converted into a heart-shaped structure like this. This caudate proembryo, right? And here the suspensor we can see like this. Here the radical differentiate and here plumule. And these two are the cotyledons. Okay, cotyledons. And finally, the mature embryo develops. And it is seen in the form of a structure like this. Here we can see the cells of the vesicle becomes narrow and narrow and narrow. Radicle here and plumule here, the two cotyledons. See that zygote, first division is unequal. This is the so-called CA. This is CB, right? Embryo proper develops from CA, and this is right heart-shaped, and this is still mature proembryo, and this is the mature right embryo having the two long cotyledons. This is the plumule, here radicle, and this is the right suspensor with the vesicle. Developing embryo is nourished by right the endosperm. This is grass embryo. Okay, it is different from the other monopods. And here, right, the single cotyledon undergoes some sort of specialization, right, and that is called a scutellum. The second one, which is rudimentary, right, is seen in the form of a small outgrowth that is called epiblast. And here the plumule and here the radicle are seen. And for grass embryo, just outer to the plumule, there is a protective covering and that is called a coleoptile. This is that flap-like structure. Similarly, there is an undifferentiated structure outer to the radicle and that is called a coleorhiza. So these are some special features, characters in a grass embryo, which is not seen in other monopods. And after fertilization, the ovary will develop into fruit and ovules become seed, right, based on whether there is right endosperm or not in the mature right seeds may be of two types. They are endospermous and non-endospermous, also called albuminous and ex-albuminous. Okay. An example for right, this is a non-albuminous seeds have no residual endosperm. Example pea, groundnut. An example for endospermous seeds, coconut, rice, wheat, maize, barley, castor, all these are example for endospermous seeds. What is perisperm? Sometimes in certain plants we can see little amount of nucellus getting persistent, and that persistent nucellus is called a perisperm, and it is seen in black pepper, beet, etc. The fruit wall is called a pericarp, and based on the nature of the fruit, whether it develops from ovary alone or some other parts of the flower contribute in the development of the fruit. Fruits may be of two types, true fruit and false fruit. Most of the fruits develop from the ovary after fertilization, and they are coming under true fruits. And false fruit example, if you are the strawberry, cashew, apple, etc. Here thalamus is also contributing in the development of the fruit. So they are called false fruit, and others are called true fruits. And that is the cotyledon, storing the food. And these are the false fruit. Okay. Apple and strawberry. The next one is apomixis and polyembryony. See, how many embryos we can see per seed? Normally it is one. But rarely in certain plants we can see more than one embryo, and that condition is called a polyembryony. Polyembryony is common in plants like mangifera and citrus. Mangifera is mango. You can see sometimes twin seedlings developing from a single seed. This is because of the presence of two embryos within the seed. Now, what is apomixis? Although seeds in general are products of fertilization, a few flowering plants such as some members of family Asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization, and it is called apomixis. Okay, so there are two processes. One is called amphimixis and other one is called apomixis. Amphimixis refers to the normal type of fertilization and the seed development, that involves gametogenesis and gametic fusion. That is amphimixis. Amphi means both, that is seed formation through two processes such as gametogenesis and that is fertilization, gametic fusion. There is meiosis, by which gametogenesis occurs, and later the gametes fuse together, and the process is called fertilization. And in certain plants we can see the formation of seeds without these two processes, and that is called apomixis. That is fruit formation without fertilization. So apomixis is a form of the asexual reproduction that mimics sexual reproduction. And there are several methods or several ways for the development of apomictic seeds. And what is the significance of this apomixis in hybrid seeds? Hybrid varieties of several of our food crop and vegetable crops are being extensively cultivated. Okay, now we have different hybrid varieties, in fruits as well as in the vegetables. Cultivation of hybrids are tremendously increased productivity. Definitely, if we cultivate hybrids, we can increase our productivity because most of them will be high yielding. But there is a problem. That problem is, one of the problem of hybrid is that hybrid seeds have to be produced every year. And if the seeds collected from the hybrid plants, and they are sown to develop the next generation, it will not show the hybrid vigor because of the process of segregation. Means hybrid seeds cannot be used repeatedly for several generations. The vigor or vitality is seen in that particular generation only, and the seeds collected from that hybrids, and if we raise another crop, we will not get the expected performance in the next generation. The reason is segregation of the characters. So progeny will segregate and do not maintain the hybrid character. So production of hybrid seed is costly, hence the cost of hybrid seeds become too expensive to the farmers. If these hybrid seeds are made to apomix, there will not be any segregation of characters in the hybrid progeny. So thus the farmers can keep on using, right, the hybrid seeds to raise the crop from year to year. There is no need for getting, right, a new hybrid seeds. Hybrid seeds are very expensive. Okay. So the one of the methods by which the farmer can use this hybrid seeds for several generations is converting this into apomix. So a lot of research is going on focusing this point of, right, within a short period, scientists will be able to convert, right, this hybrid seeds to apomix, by which we can enhance our productivity. Okay. So with this, the chapter is over. Thank you for watching.